In this section of notes, we're going to tackle Unit 4, which is all about the cell membrane. And I know we learned a little bit about the cell membrane in the last unit. What we're going to focus on today, that's highlighted in pink, is we're going to look at what makes up the cell membrane, so what molecules make up the structure, and we're going to take a look at some of the functions of the cell membrane. First, on page 1, we're going to focus on some functions. We've already learned that the cell membrane regulates what moves in and out of the cell. So let's draw that some molecules can move through the membrane, but note that not everything can move through the membrane. We have a term for this. Since some molecules can move through the membrane and others can't, then we say that that membrane is semi-permeable, or the membrane is a semi-permeable membrane. Another way that we can say that is it's selectively permeable. These both mean that some molecules can move through them and some things can't. We're also going to talk about how the cell membrane helps you maintain homeostasis. If you're maintaining homeostasis, what you're doing is you're maintaining a constant, which means unchanging, internal environment. So what we're trying to indicate is that you're trying to maintain certain ideal levels. Like you have an ideal temperature, you have the ideal amount of water that you should have, the ideal amount of certain solutes that you should have in your system. And your body and your cells are always working to try to maintain those ideal levels, or you're trying to maintain homeostasis. So for example, some things that you're trying to maintain is your solutes inside and outside of the cell, and also the water that is inside and out of the cell. We're going to talk about other things that you're trying to maintain. Another function of the cell membrane that we haven't discussed yet is the cell membranes are key when it comes to cells communicating. So it allows cells to communicate with each other. Now in class I show a short video clip that gives you an idea of how cells can receive signals. Signaling molecules that are made in one part of the body and released to move to other parts of the body are called hormones. This hormone is a signal. The signal can bind to specific cells in the body, and it can cause them to respond. So cells aren't communicating using sound, they're communicating using chemical signals, which we commonly call hormones. So they're communicating through these signaling molecules that we can call hormones or ligands. And again, hormones are produced by cells in one part of the body, and they're going to travel to target cells in another part of the body to cause them to respond. Now, not only can hormones bind to proteins on the cell membrane, but other things can as well. For example, toxins can bind to cells, and if they're binding to white blood cells, then your white blood cells are going to respond and make more white blood cells that are going to take out the bacteria that are producing those toxins. Viruses can also bind to the membranes of cells, and this could be a good or a bad thing. If the viruses are binding to white blood cells, that means that the body's detecting the viruses, and they're going to produce more white blood cells to kill the virus. But sometimes viruses bind to the structure of cell membranes, and that's what causes the cells to take the viruses inside, and now you have cells that are infected by a virus. So there's many things that can bind to the surface of, or the cell membranes of cells, and can cause them to respond. So cells can communicate using structures in the cell membrane. Now let's take a closer look at some of these structures. We've already mentioned that the cell membrane is primarily made of phospholipids. We talk about that in the biochemistry unit and also in the cell unit. In addition to phospholipids, though, we have these proteins that have different jobs, and they float around, or we say they're embedded in that phospholipid bilayer, and also cholesterol. That's a lipid. Cholesterol is important in cell membranes, so we can't lower our cholesterol too much because we do need some cholesterol to build those membranes. Now, how these molecules come together to form that structure that you picture when you picture the cell membrane is due to the fact that these molecules have polar and non-polar properties or regions, and that's going to affect how they interact together to form this barrier that's on the outside of the cell. Remember, those phospholipids have polar heads, and the tails are fatty acid chains, and fats are non-polar. If you remember, since the head is polar, polar will interact with water molecules, which are also polar. And the head of the phospholipid, since it's attracted to water, we say it's hydrophilic. So think of that as meaning water-loving. The head loves water. Okay. As for the tails are non-polar, and they won't interact with water molecules. The tails are hydrophobic. So think of that as water-fearing if it's hydrophobic. Now, because of the polar heads and the non-polar tails, and the fact that on the outside of the cell you have water, and in the cytoplasm you have water too, the phospholipids on their own will arrange themselves in a bilayer so that the polar heads are forming hydrogen bonds with water molecules, and then the nonpolar tails are interacting because nonpolar molecules want to interact with nonpolar molecules. And those nonpolar tails won't be interacting with the water. So we end up forming this bilayer, which makes that cell membrane of a cell. So phospholipids arrange themselves into a bilayer because those polar and nonpolar properties of those phospholipids. Now, the proteins also are going to have polar and nonpolar regions, and same with cholesterol. We're going to focus on the functions of the proteins on the next page, but let's talk about why cholesterol is so important. As we mentioned, your cell membrane is mostly made of fat, and if you heat up fat, fat will liquefy. Well, that would be bad if your membrane liquefies, because then it wouldn't be able to keep the structures inside of the cell. Also, when fat becomes too cold, it becomes solid, and as an animal, we need to be able to move around, so we need very flexible cell membranes. So what cholesterol is able to do is it stabilizes the membrane when temperatures change. It keeps the membrane more fluid when the membrane or the cell is cold, but it keeps the membrane more solid whenever the temperature gets too warm. Again, we wouldn't want that cell membrane to liquefy. Now in class, we built a model of the membrane on the board. Students were given proteins and cholesterol molecules, and they had arranged them into the phospholipids. So let's make sure you understand what they had to understand before they could do that. Remember, we said that the phospholipids are polar, and since there's water outside, those heads will face the watery outside environment and also the watery environment that would be the cytoplasm. But let's focus on the proteins here. This part of the protein, since it's facing where the watery environment is, that part would be a polar area of that protein. Whereas this part here that's around those phospholipids would be nonpolar. I'm just going to put NP for nonpolar. So you can see that even proteins have polar and nonpolar regions, and so does cholesterol. This part up here is a polar part of the cholesterol molecule, where these parts down here would be nonpolar areas of that molecule. Now, how these molecules come together is a model that we call the fluid mosaic model. We're talking about the fluid mosaic model. We're always going to be talking about the cell membrane. One, that the cell membrane is very flexible. If I was to hit it, this would kind of wave like the surface of water does. And mosaic means that you have lots of pieces that come together to form this final structure, like a mosaic piece of art. Now, some other things that you need to understand is what we can call some of these proteins. If I'm referring to a peripheral protein, like this one right here, peripheral means on the outside. So this is a protein that's not actually inside the membrane. It is on the outside of the membrane. And we have then other proteins here, which are called integral proteins. And integral means inside. So the fact that you have these proteins embedded onto the inside of that cell membrane makes them integral proteins. Hopefully it makes sense why we would call this protein an integral protein, because it's actually inside the cell membrane. But also it's called a transmembrane protein, because trans means across, and you can see that this protein goes all the way across the cell membrane, connecting the inside to the outside. When we look at this protein here, this glycoprotein, it is also an integral protein or integral protein, but it's not a transmembrane protein because it doesn't go all the way across. Now, in this unit, we're going to be creating a student-generated test, and there's a handout that shows you're going to answer, or you're going to create 10 questions, and each question has to have a diagram that students would have to analyze in order to answer that question. And from what I've covered so far in your notes, you should be able to write a question for topic number 1. Topic number 1 states that you're going to write a question to determine if a student is familiar with the internal structure of the cell membrane, such as the arrangements of the phospholipids and the proteins and cholesterol, and why they would be arranged like they're arranged. So you need to probably ask questions about which areas are nonpolar, which areas are polar. You can also ask questions about transmembrane protein versus peripheral proteins. Like, why would peripheral proteins be on the outside of the membrane instead of on the inside? Again, you have to include some kind of diagram that must be referenced before anybody would be able to answer your question. Now, for topic 2, we're going to focus on this next page in our notes, the functions of the different proteins that are embedded in the cell membrane. So we see that we have phospholipids with these proteins, and these proteins can have different jobs. So there's 4 main types of jobs that we're going to focus on. First, we have transport proteins, and this is pretty easy. Transport proteins transport molecules across the membrane, either into the cell or out of the cell. Now, we can call these transport proteins by different names. Sometimes we say these transport proteins are channel proteins. So we can call them channel proteins. Sometimes they're called carrier proteins or carriers. And sometimes we might be talking about pumps, and we're talking about transport proteins that are pumping molecules across the membrane or moving molecules across the membrane. So there's just some other names for transport proteins. Now, marker proteins are going to have a couple pieces to them. We have the protein part. Let's indicate we have the protein part that's embedded in the phospholipids. But then you have an additional part to it that oftentimes is diagrammed what looks like a chain. And that is, in fact, a chain of glucose molecules. Now, if you remember, glucose is a carb, so let's indicate this part is a carb chain. It's made of glucose. Remember that we take a bunch of glucoses and we string them together in our body, then that can make glycogen. That is why these marker proteins are often called glycoproteins. That just simply means you have a protein that has a carbohydrate chain attached. Now, the job of these marker proteins is to identify the cell type. We can identify cells just based on the proteins that they have on their surface, or their marker proteins. Now, these can be good things and they can be bad things. If you needed a kidney and someone donated a kidney to you, your body wouldn't recognize the marker proteins on those kidney cells and will spend your entire life trying to destroy them. So your markers are different from other people's markers. And when we're trying to match someone to a donor, what we're trying to do is match marker proteins. We're trying to find kidney cells that would have markers similar to yours, so maybe you don't have to take so many immunosuppressing drugs so that your white blood cells don't wipe out your donated kidney. Also, your different cell types are going to have different markers. Like, for example, your white blood cells, so let's put white blood cells, they're going to have different markers on their surface than your skin cells, or your red blood cells, or your liver cells, and so on. Now, receptor proteins, we've already mentioned them, they can receive signals. If you remember, we talked about cells can produce hormones, like right here it's showing you this would be a hormone that has bound to this receptor protein, and you can see right here that there's some kind of signal that is happening that can cause the cell to respond. So receptor proteins receive signals or receive hormones to cause a cell to respond. And then we've already talked about enzymatic proteins. Remember, enzymes perform chemical reactions for the cell. Now, you can have enzymes in the cytoplasm doing reactions for you, but you can also have enzymes that are embedded in a membrane. So they're not moving around. They're staying in a membrane. Remember that enzymes can take one molecule. So X would be the substrate in this situation. And you can tell that X is being turned into Y. So Y would be the product. And that's why we would call this protein that's embedded an enzyme or an enzymatic protein. So again, these are the 4 functions of proteins that are embedded in the membrane, and that's what your question 2 topic is going to be about. Remember, you have to provide a diagram that must be addressed for a student to be able to answer your question on question 2 of your test.